Okay, for the state government, say no, but then for different states, there are different laws. And yeah, so what do you think would be the problem? Not the problem, but the underlying ideology or thinking affecting this law in this regard? Why that some countries are more supportive, some countries don't? We all know that in Europe, Switzerland is always supportive, right? Because they have a very strong biotech industry. Many people go there to do injections of hormone and make them look young. But to me, that would be very awful if I'm 60 or 70 years old but still look like 25. Right? I would find this so scary. You see what I mean? You're still walking like this, but then you look, oh, this man or woman actually look very young. So kind of, give me one reason why you don't support it.
Religion.
Hmm?
Religion.
Religious?
Yeah. Like some religions believe that the embryo is alive, and it's like conceived and not.
Yeah. So you can see that many advanced nations, including Japan, Britain, they become supportive. I suppose mainly because in this country they have accommodated different religious beliefs, right? Even in Britain, or actually in UK nowadays, there are different religious bodies. In China, obviously, religious is only to communism, only. Okay? But then in other countries like Austria or Germany, they suffer also from what? In the history of political depression by other ethnic groups. Okay, for those people, they don't really support manipulation of human, because that would relate to what? Eugenesis. Okay, 要上学. So those countries, they obviously don't like to see that to happen. So gene editing, coming on to this last part, everything changed. We're expecting the scientists discovered CRISPR-Cas9 technology to win the Nobel Prize someday, but they're still debating on their discovery, on the patent. Originally from bacteria, this is the immune system of bacteria. We call it the immune system of bacteria because any incoming DNA, remember 50 years ago we've discovered restriction enzyme system, right? The restriction enzyme would come out and chew off DNA. At the same time, in the bacteria genome, not just the restriction enzyme, but also they have the methylation system to protect its own genome, right? So we thought that, okay, that is it. That is a very good protective system or mechanism to protect the bacteria from incoming invading bacteriophages. But actually it's more than that. It is believed that not just bacteriophages, but also many other DNA are coming into invading the bacteria. So the bacteria actually developed this CRISPR-Cas9. There's a very long name, but we usually call it CRISPR. And so scientists actually could use this technology to manipulate or insert any DNA into its target genome. Okay? So the technology originally from, not technology, but the mechanism originally discovered in bacteria. And the CRISPR usually come with Cas9, and before that there are different similar mechanisms known as like a TALENT, T-A-L-E-N-T, and also other technology. It relies on the base pairing of the DNA to find the target DNA, and then the target DNA would guide this enzyme. The enzyme is known as Cas9. In other words, it's different from the restriction enzyme system. And this enzyme is kind of like an enzyme that would digest anything based on base pairing. The base pairing is guided by this CRISPR. So this is the CRISPR design. You may design this. This is fixed because this is for Cas9 enzyme. and this is the target, and no matter any target that you want to add in, you may just do gene synthesis and synthesize the DNA, and then you may kind of guide this CRISPR-Cas9 to cleave any target DNA. And it works like a charm. It works in different cells, in human cells, in cell culture, it works in bacteria, of course, and it also works in zebrafish very well. Not just zebrafish, but also mammals, including mice, okay, rats. The embryos could be acted like that. Of course, they not just not done something, but also at the same time to use the genetic recombination technique to introduce the new gene. But step one is to remove the gene that they want to repair, and then step two, they would like to add the gene in. So the overview of the genome data, actually this is the announcement came from, I only cut one slide from Dr. Kerr's discovery or invention. What they did is that they have an HIV carrier father and an HIV negative mother. You know, the expertise of Dr. Kerr or Kin Fui is actually a genome scientist. We do have many genome scientists actually in our school as well. Teng Fong was just talking to me that the students already open companies in Science Park and trying to do a whole genome sequencing for individual patients. Okay, so the technology now is so simple that you may do cotton swab and then collect the genomic DNA and do genome sequencing of individual genes, of course. But in this case, Dr. Kerr is an expert doing all this genome sequencing. So peripheral blood genomic DNA prepare and then could do a single sequencing. So that would identify all those, they call it de novo, idols or halo phase, to study all the different major genes in the genome of these individuals. Now the question here is that after fertilization, they prepare the eggs. Before it goes into blastocyst, they did Cas9, CRISPR injection. So they inject the guided RNA and also inject the Cas9 gene to express the Cas9 enzyme to edit the embryo. Okay, so and after that they also did sequencing to confirm whether the editing was successful or not. And then after that they prepared the fertilized egg after editing to back to the mother, and that mother gave birth to twin, Lolo and Lala, Lulu Lana. Okay, and again Lulu and Lala will over the time during the pregnancy. During the pregnancy, they also did sequencing. Okay, so all of the major funding actually went to this genome sequencing. Every single step they sequence the whole genome. There are major concerns here because you never know where of course it's guided by this specific genomic RNA to guide the Cas9 to do editing, but you never know when. The time is critical here because the embryo is developing, right? At the same time you inject the RNA for the guiding and also the DNA for the expression of Cas9, but you never know when the exact timing of editing happened. And we usually call this mosaicism. Mosaicism means that then the time that after or during embryogenesis, the different cells start to move around and divide, right? Not all of them got added. Not all of them got added. Because it's a critical time that shortly after fertilization or as soon as the egg is fertilized, you should have injected this in. But even so, after the DNA and Cas9 injected, you don't know when it could get into the nucleus of fertilized cells divided into 2 or 4 or 8. Which particular cells got added? We don't know. And therefore, they got the time to check all the DNA samples over 12 weeks, 19 weeks, and 24 weeks, and so on. And then at the time when they were born, and of course, according to them, they have found the evidence of gene editing, preparing Lulu and Lana to have special genes to deal with HIV. Okay, of course, one of the key issues that is on debate is whether the manipulation of baby is allowed. Now, he actually showed us this from the hospital, but we all know that. I still remember that day we all received the WhatsApp, and then we asked our other biochem colleagues, oh, no, suppose the Ministry of Health of China actually banned all those different steps, right, I just mentioned in previous slides. So how come they could develop this medical ethics committee approval? Okay, it's either unauthorized, or this is not a real document, or they have done something wrong. Okay, but you would imagine that if that is possible, the technology actually right now, CRISPR-Cas9 technology could be applied into different steps. Good for animal models, good for genetic studies, good for develop into different material for food production, for fuel production, for gene therapy, and so on. But not for editing a baby. So I mentioned again, so this is the guy who did the work, and now he disappeared. Okay, 1 week after he made the announcement, he was found in his residence in Shenzhen, and then nobody heard anything from him again. All right, so that's what he made the presentation last year, December, in Hong Kong U, a conference organized by the Academy of Hong Kong and the National Academy of Medicine and also the Royal Society. And then he announced that they have edited baby, and then the baby Lulu and Lana, and then they found that the target gene's been edited. Okay. The question number 2 is that we are not so sure whether the edited genes is beneficial to avoid HIV infection. Now that's question number 2. Many scientists have query on whether this mutation could make Lulu and Lala become unaffected by HIV. Now that was his goal, right? Trying to edit the baby without being attacked by human immunodeficiency virus, HIV virus. Okay, that's question number 2. So at the end, editing baby could be wrong in any cases, as I mentioned, in mosaicism, a one-cell embryo and then different cells. So how do you know when the gene get in? The problem arises here, abortion or high-risk pregnancy. So after editing, would you know that the target gene, how many copies of the genes got in? Okay, and unknown embryo genome sequence after in vitro fertilization, we are not so sure at the one-cell stage. They should remove the genome for sequencing, right? But if you remove the genome for sequencing, there will be no embryo. So you cannot confirm that stage. And then what we call next step is the off-target effects. Because the human genome is rather complicated, much more complicated than bacterial genome, right? So how would you know other effects which are not known to human? Okay. And then the wild-type embryos, mosaicism is upon here. Throughout these developmental stages, how would you know the changes on the germ line or these different cells? In many cases, it's related to germline clinical use because many of these cells are developed into different organs. So you're not so sure whether the editing has been successful or not. Of course, you will say, okay, we better wait for the baby to grow up to do more analysis. But unfortunately, now it's a big black hold，完全係一個黑洞。Nobody know exactly where is Dr. Kerr. Nobody, I haven't heard about, actually Dr. Kerr had a collaborator from Texas. Okay, I keep contacting my friends in Texas and ask if that person been gone back to the US or still in China and so on. Because that person is also his supervisor, Dr. Kerr's supervisor is also facing similar charges in Texas. Okay, of course, you say, okay, I only did that in China, not in Texas. But still, at what stage the technology been involved? All right, and then nobody know exactly what happened to other pregnant women, because according to Dr. Kerr, the announcement were only the successful cases. According to him, there are other survivors in the experiments waiting for further analysis. According to Kerr, he signed the agreement with many other people or fathers to do these experiments, okay? So there are bioethical concerns, including number one, we cannot do any clinical trial. Even clinical trial, we need further different steps before you can do clinical trial, even for drug testing. So not to mention gene editing. Number two, Lulu Nana had not been agreed to be added. Now this is a very difficult issue. You know, the individual could never be consulted, right? Only the parents. But then the parents, because their treatment and then the experiments actually not justified. We don't know whether the editing would damage other genes. So how would you believe that the parents had made the right decision? The parents, even the parents for genetic counseling, in many countries it takes a very long time to do what we call the genetic counseling, only for normal genetic, right? I mean for genetic diseases. We're now talking about baby editing. So the parents involved, they've been told the consequences. The matter of truth is, even the scientists don't know the consequences. How could we do any consultation or counseling? So of course we need to do more experiments on embryonic stem cells and to make sure that the technology is available. And also coming back to the original thinking, step number one, do you think baby editing is ethical? So with that, I think if you want to know more about the whole story, you may click into these videos or forum. And to many scientists, they think that would be good, but Dr. Kerr perhaps went too early, perhaps 50 years early. All right? I don't know. I still have questions for you. Whether gene therapy is ethical, gene editing is ethical, designing baby is ethical, or even baby editing is ethical. Now, and then number 5, what's wrong with Dr. Kerr? So a little bit longer than 10 minutes long. So we take a break and then we come back and we have discussion on that. I'll explain to you how are we going to discuss this topic. Okay, let's take a break.